China has identified the cause of the
mysterious new virus,
coronavirus.
What is the origin source for COVID?
The first U.S. case has been detected.
There are fears a rapidly spreading virus has reached Australia.
Mainstream media has been pushing a narrative of one type. It is solely a bat coronavirus that escaped from a cave in Wuhan and has infected the entire planet. I didn't know there was a different narrative.
I have today declared that the coronavirus presents a public health emergency in the United States.
And I couldn't believe that in December of 2021, that in January of 2020, there were all these articles, even in CNN Health and Healthline Online, medical journals, that geneticists in China in January of 2020 had already determined that the origin source of COVID was two snakes.
Also at 10, snakes starting to play a big role in COVID-19 research. Scientists from the U of A discovering an enzyme similar to one found in snake venom could be driving COVID death.
What they had found is that in a 30,000 coded sequence of SARS-CoV-2, they isolated really short chains that matched venom.
Researchers in Italy took COVID-19 patients from multiple cities, and then they took 10 people from multiple cities who tested negative PCR-wise for COVID. They took their blood, urine, and feces. They found a total of 36 different animal venoms. My question is how did 36 different venoms end up on land inside the tissues of COVID-19 patients?
In the course of the investigation, CIA's laboratory storage facilities were searched, and about 11 grams, a little less than half an ounce, of shellfish toxin and 8 milligrams of cobra venom were discovered in a little-used, vaulted storeroom in an agency building.
Mainstream media has been pushing a narrative of one type in relationship to SARS-CoV-2 and the COVID-19 pandemic as a whole, which has been, it is solely a bat coronavirus that escaped from a cave in Wuhan and has infected the entire planet. That was the accepted narrative this entire time. And my entire focus for the first 2 years of the pandemic was to protect people from going into hospitals and considering early treatment at home that had therapies, maybe prescriptions they might need that everyone had access to that could save their life and never have to go to a hospital. Dr. Richard Bartlett and I have shared stages for 2 years now. He is basically known as the budesonide guy. He was the doctor that came forward, went viral in May of 2020, that you don't have to be scared of COVID. Budesonide, this nebulized inhaler for asthma for preemie babies up to 100-year-olds, can cure COVID.
Continuing coverage tonight on a controversial COVID-19 treatment. The inhaled steroid budesonide is being hailed by West Texas Dr. Richard Bartlett as a silver bullet to treat COVID-19. Venom Memorial Hospital isn't convinced, telling us yesterday there's no such thing as a silver bullet for the virus. But Bartlett's patients credit the treatment for saving their lives.
When it comes to COVID-19, Kathy Lawler and Eric Rodriguez are at risk.
I'm 48 years old, I'm diabetic, I'm a bit overweight.
I am in my 60s, which is already a strike against me, and I am living with 2 different types of non-Hodgkin's lymphoma. In fact, I'm on daily chemo.
When they were diagnosed with the virus, they both thought worst case scenario.
Scared, desperate, fearful. I started thinking about my children. Without a father.
Rodriguez lost his 31-year-old brother to the virus in June.
He said he couldn't breathe very well.
And for that reason, he sought a different treatment than his brother. He started taking budesonide twice a day.
I think the difference is life and death right now. I feel fantastic.
As for Lawler, well, she believes it would have been a different outcome if it weren't for Dr. Bartlett's regimen.
And I fully believe that by the time they decided I was COVID positive, had I waited on treatment, I would have been in the hospital on a ventilator. Trying the budesonide, I could tell improvement. I could feel that something was changing.
I didn't know there was a different narrative. I was just going along trying to protect people's lives from ending up in a hospital. Well, I got this text from Dr. Richard Bartlett, and it stated, Dr. Artis, if you got bit by a rattlesnake, would you go to a hospital and get anti-venom? This shifted the entire narrative of COVID for me.
What I looked at from the beginning is that if they were saying that this was SARS-CoV-2, right, and originally when they called it Wuhan, and then originally when they said, is this from a bat? Then I started looking at information around SARS, MERS, right, and making comparisons. And what I found very quickly is this was SARS. This was SARS-1. Dr. Brian Fry, the world's expert on venom, said, oh, there's nothing weird about SARS-CoV-2. It's in the same family as the rest of its family. And then I also went back and I went, there's something else weird. If they're saying that this is an acute respiratory issue, then why am I seeing these videos coming out of China and out of Wuhan where I'm seeing a nerve gas attack? What is happening where I've been around hospitals and patients, and most people have had someone in their life that's had a bad pneumonia or a flu. You've never been around somebody that suddenly goes into a seizure on the floor from getting a respiratory virus. Maybe that becomes the end point. You've never seen somebody suddenly drop dead in a street without any other symptoms leading up to that. They're walking across the street with their friend and the next thing they're in the street. And we never had that in the States, right? If we were truly following this biological, this is just from a bat, we're still looking for the animal. Is it a pangolin? Is it a snake? Is it a bat? Is it a rat? This game that they were playing meant that there was a distraction of, if we have the same problem that Wuhan has got, why aren't we seeing the same symptoms?
Richard Bartlett had an infusion center he had open for 4 months where he was treating acute COVID patients, outpatient, simply with monoclonal antibodies called Regeneron. And he had 100% success at curing people with COVID using monoclonal antibodies. So he sent me this text in the beginning of December, knowing I didn't know something about monoclonal antibodies. I didn't know that's what the text was about. It just said, hey, Dr. Artis, you got bit by a rattlesnake. Would you go to a hospital and get anti-venom? And I knew him as an ER doc must know something about anti-venom. I don't know, as a retired chiropractor. So I typed online, what is anti-venom made from? And I couldn't believe it, that 90% of all anti-venom around the world is monoclonal antibodies. Then I did realize what he was trying to get across to me is he knew me as a researcher. I would dive in to find out what I didn't know about anti-venom, and I would come to my own conclusion, which is his conclusion. If monoclonal antibodies are used as anti-venom therapy for snake bites around the world successfully, why is monoclonal antibodies almost 100% effective against the coronavirus? Why is this anti-venom therapy called monoclonal antibodies working against a bat virus? So I went online immediately and I went like this, what is the origin source for COVID? That's all I wanted to know. I actually typed in, what is the origin source for SARS-CoV-2? And I couldn't believe that in December of 2021, that in January of 2020, there were all these articles, even in CNN Health and Healthline Online, medical journals, that geneticists in China in January of 2020 had already determined that the origin source of COVID was 2 snakes, the Chinese crate snake and the Chinese king cobra. The third most likely animal reservoir source for this virus was bats. I couldn't believe that. And then everyone around the world was publishing it, that this was a respiratory disease from snakes.
I came across this paper around, it wasn't even necessarily around venom, and it wasn't necessarily around SARS. This paper explains that it looks at different gut circumstances, and as it relates to what people would say is sort of the connection between the immune system and the gut and the brain, and it looks at sort of gut health. And then buried in this paper was probably purposefully so that the paper could still get through peer review and challenge the narrative, was the section that said, we've analyzed SARS, and this is the genomic sequence, and within this genomic sequence, we've isolated these 2 toxins. And then they run it just like I did to confirm it. you run those sequences through something called BLAST. What they had found is that in a 30,000 coded sequence of SARS-CoV-2, they isolated really, really short chains that matched Venom. Then they took it one step further in this paper, and they said, these two venoms, the closest match is Cobra and Crate. And then their science was amazing, and they didn't call it Venom. They said they are homologs, meaning they are copies. Because these two sequences are there, everybody's focusing on ACE, these ACE receptors. But there seems to be a very high likelihood that these two sequences are binding to these things in our bodies called nicotinic receptors, acetylcholine nicotine receptors. And they said, isn't that part of the reason that there's pathology taking part in the body? And it was the fact that there was even a cartoon, what they call a cartoon representation. Like, I'm a picture guy. And so when they said, here's the section that's Cobra, and here's the section that's Crate, I didn't need any more. Unless these were liars, and unless this paper was suddenly going to get retracted as, oh, we made a mistake. They just spelt it out.
Not only was it confirmed in China in January of 2020, researchers there took people that were sick in Wuhan originally in the fall of 2019, took their blood looking for antibodies. When they isolated the antibodies that were fighting the disease process they were in, which they called a novel pneumonia, when they took those antibodies, they analyzed the genetic sequence, and the origin was two snake venoms that the antibodies are directing the attack against. And it was the Chinese crate venom and the Chinese crate snake. 4 months later, in April of 2020, imagine my shock that there's French geneticists who have isolated the spike protein on the outside of the SARS-CoV-2 virus all by itself, ran its gene sequences, and they published their findings that it's two snakes' venoms, peptides, short sequences of proteins from the venom of Chinese crate snakes and Chinese king cobra once again. And then they also published that there's 3 genetic sequences of rabies virus also that can be identical to this spike protein. And then I'm looking for confirmation that did anyone else find actual venom in patients with COVID? And in July of 2020 is when researchers in Italy took COVID-19 patients from multiple cities. They took 20 of them that PCR tested positive for COVID, had symptoms of COVID. And then they took 10 people from multiple cities who tested negative PCR-wise for COVID and had no symptoms. And they took tissue samples from them while they were either sick or just a control group that wasn't sick at all. They took their blood, urine, and feces, and they did what's called liquid chromatography and mass spectrometry, which are just big fancy machines that look for stuff in tissue. And they ran all the tissues of the 20 COVID-19 patients, 10 control members from multiple cities. They found a total of 36 different animal venoms in the blood and the stools of only the COVID-19 patients, and found 0 in the negative COVID-19 patient group. What was interesting was this study has been peer-reviewed, approved, and published. And you can find it online right now. So they confirmed that there was 36 different venoms in the blood and feces of COVID-19 patients only. And they include the crate snake, the Chinese king cobra snake, a spitting cobra, coral snake venom, brown snake venom, moccasin venom, Chinese water moccasin venom is in these patients. And they also isolated 16 different venoms from a starfish called the crown of thorn starfish, which is a deadly venomous starfish in the ocean, in blood and feces of COVID-19 patients. And they found 15 what are called conotoxin venoms that are also identical neurotoxins to the snake venoms they found. And those are snails found in the ocean called cono snails. My question is how did 36 different venoms end up on land inside the tissues of COVID-19 patients?
I don't have a bias in me. I don't have an ego. I don't have a dog in this fight to say, oh, this is nonsense. It can't be venom. I just said these scientists found venom. And then I like to be very methodical. So just like them, they said it's crate-like, but they left room for the fact that they couldn't quite work out this crate sequence. But the cobra was almost a perfect homolog. And so I spent one night teaching myself how to use BLAST, and I run the sequence. It came out as Cobra. And that was enough for me to then move on and have that sort of aha moment. But it wasn't like, oh, wow, I've just discovered. One, it wasn't me discovering it, right? It was this amazing group. And then there were other papers around the same time that were coming to similar conclusions. So the weirdness around the rest of the S protein is the defining factor for me. When I started looking at it, and I was very quick to understand that the S protein was the pathogenic profile. By the time they then isolated and started working on the spike protein, there was an experiment that I found where they took the spike protein and they introduced it to tadpoles. So when they took this substance, right, which I've now and other scientists have identified as a venom and as a toxin, people are uncomfortable to use the word venom, I'm sure. You know, go to your safe space, think about it for a while, come out of your safe space, and then choose your preferred, just like people want to choose a preferred pronoun. I don't care what you call it. There's this thing that causes death, right? And so you have these poisoned arrows, because that's the shape of them, right? And they're soft, and well, they soft, but they're also metal, so that's also weird in terms of it kind of is a nanoparticle arrow. And go and put it in a jar and go and see what happens with the tadpoles. And what happens with the tadpoles is an envenomation. So not only are you now seeing the genomic sequence, and let's say you didn't believe the genomic sequence. Let's say you couldn't understand the genomic sequence. This would be the equivalent of the most basic experiment. Could this thing over here kill this thing over here? Okay, I'm going to take this thing over here, I'm going to put it in here, and I'm going to watch what happens. Then it kills these things or changes enzymes. And the enzymes that it changed and the effect that it had showed that this is potentially cardiotoxic, but more importantly, it absolutely proved that it was a neurotoxin.
A lot of people right out of the gate, when they learned about my discovery and narrative of weaponized venom, calling it COVID, a lot of people immediately thought there's no way that there's enough snakes to milk venom to make 8 billion people on the planet sick. And that is very true. All I said was they're using snake venom peptides as a weapon. And what people don't know, and that I did not know that I had to learn, I did not know that since the 70s, 1970s, we have been mass producing in factories, biological weapons labs, and in pharmaceutical drug factories. We have been making synthetic venoms of these marine snails since 1979. We've been making synthetic king cobra venom since 1975. We are making massive amounts of synthetic snake venom peptides that are being used in cancer research, cancer treatment, drug therapy, drug modalities, drug creation. Lots of them come from snake venoms. There's over 100 venoms right now being used, isolated, and in research right now to be isolated and be turned into drugs that human beings will either inject into their body or swallow. So this is a massive field I didn't know existed. Lots of snake venom peptides are artificially and synthetically made, and they're just as biologically toxic and dangerous as the actual venom. They're just synthetic versions. It makes sense to me that they're actually just weaponizing venom. I don't even think you need a virus to spread this. I think my actual take was, I think they're taking these snake venom peptides that are actually lyophilized, freeze-dried powder. Once they get these synthetic venoms, they actually look just like baby powder, and it can be put in anything. And my worry was that they're actually putting it in water and we're ingesting it. And then that's how people are getting sick with COVID. My actual what I was relating to the world was, my concerns is that they're poisoning the water with venoms. It's appearing to be a respiratory disease because the venoms, it doesn't matter if they get in through the gastrointestinal tract or you breathe it in through your lungs and get it into your blood. The moment it gets to the blood, it will cross the blood-brain barrier. All these venom peptides do. And they attack neurological tissue in the brainstem that controls respiration. That's how venomous creatures kill their prey. They paralyze their body's ability to breathe by getting to the brainstem and hitting specific receptors called nicotinic acetylcholine receptors. And these venoms of the krait king cobra and those 36 that I mentioned that were in the Italy study all cross the blood-brain barrier and get to the brain. It could be either it was released in the air or it's in the water and we're drinking it.
I took this information of working out that the issue is that the spike protein is the driver of the disease. And immediately for myself, and stop wasting time, right? I said, okay, so now the suspect of concern is the spike protein, and I discarded any research or anything on viruses. The spike protein contains venom, it contains HIV inserts, it contains things that are basically the same inserts as rabies, and it also has what they call a super antigen bacteria. Immediately when they said that the S protein is the basis of their vaccine, right? Now, immediately that, you don't have to be a virologist, a vaccine developer, you don't have to be a scientist, but everyone knows that the basis of vaccines is to create an effect of immunization by taking a substance that's toxic, making it innocuous in some way, attenuating it, making it dead, attaching it to something, making a copy of it that's not, that's similar. In a sense, all vaccines are a form of homeopathy, which doctors would put down homeopathy. But what vaccine is an attempt to create a resonance in the body that the body says, hey, that's a pathogen, I'm going to make a response to it. But the pathogen's not there, right, in that form. But when they are telling you that they're not using the virus and that something's different about this, beyond anything to do with mRNA technology, I knew that AstraZeneca is going to be a problem, J&J is going to be a problem. The only one that I knew was potentially not going to be a problem was Sputnik. Because who in their right mind wouldn't know that Russia's trolling you, that there's no way they're about to kill their own population? And what better way to show that you're not in on the joke? By calling your vaccine Sputnik, right? You're like, in your face, we're not along with this, okay? And our vaccine is going to be so laughable that it's more than likely placebo, and we're going to show you how laughable this is, that our vaccine, it doesn't contain the word cobra like Corbavax out of India. It doesn't have this new technology in it. We're just going to call it Sputnik. And so the other vaccines, the minute, whether it was mRNA or not, the minute they are putting the weapon inside a vial, and I know that that weapon is venom, now it goes from the discomfort that biologists have, where like, doc, you can't call this venom. Venom needs envenomation. You need to fit the description that it needs to bite, or it needs to sting, or it needs to inject itself, or you need to stand on the sea urchin, or you need to get stung or bitten by the coral snake. And I could even bite my tongue and say, that's not quite true, because a cobra aerosolizes its venom, and it sprays it out of its mouth, and it goes to work without biting anybody.
Not only did I have a concern that they were weaponizing venom, calling it COVID, based on these scientists' peer-reviewed, published research papers. Not only was that convincing, I then was worried that all of the adverse events being reported from the vaccines, that those also could be tied to venom. So what I did was I wanted to know, is there anything in relationship to the COVID-19 vaccines I've never heard before that I could then go research that possibly there's a connection that I might be right in my inclination. A medical doctor out of British Columbia, Canada, named Charles Hoff, he had all of his patients were vaccinated using mRNA vaccines. I believe it was the Moderna shot. And when the Moderna shot went into all of his patients, a lot of them started complaining of long hauler-like symptoms: fatigue, exercise-induced lethargy, brain fog. So he ran a blood test on all of his mRNA vaccinated patients, and then he reported to the world that he's seeing 60% of all of his vaccinated patients with mRNA COVID-19 shots had elevated D-dimers. And he said that means there's massive amounts of blood clotting going on in the body. I, in my interest to see if I could understand a correlation between venoms and vaccines for COVID, I went online and I typed in, how are medical doctors and lab technicians trained to look for the causes of elevated D-dimers? Because I didn't know. I've never even heard of this test. So I looked up online, and there's a great online medical journal called Medscape. And the very first link that pulls up when I type that information in for an internet search, it came up with an article from 2019 on medscape.com. and it describes that elevated D-dimers represents blood clots. But this is, the title of the article is How to Interpret Elevated D-dimers. And it states there's 5 things that every medical doctor should be looking for when you see elevated D-dimers, including Dr. Hoff in Canada, which he was right about. The first 4 describe various types of blood clots in the body, either deep vein thrombosis, pulmonary embolism, micro blood clotting, and then the 5th bullet point is the only one that actually has a causative agent that could cause the other 4 blood clots described, and that is snake venom poisoning. Well, if Charles Hoff, MD, in British Columbia, Canada, was reporting 60% of his mRNA patients who got injected have elevated D-dimers, and medical doctors are supposedly, per the medical literature, told to look for snake venom poisoning as a cause for elevated D-dimer, all I wanted to know then was, who created these shots, and do they do snake venom research? That was it. So I went online to find out who actually created these shots, and to my shock, it wasn't Dr. Robert Malone, it was actually Dr. Drew Wiseman and Cataline Carrico at the University of Pennsylvania. They are the co-credited inventors and patent owners of the mRNA COVID-19 technology and receive royalties on all of these shots, and have received Lifetime Achievement Awards for saving the planet. That's these 2 people. I never even heard of them. Once I knew their names, I just copied their names, put it into an internet search, and I said Drew Wiseman and Cataline Carrico and snake venom research. I was shocked that when I hit enter, there's a list of research studies that come up immediately, starting in 2009, where these 2 people are using snake venom in their research. The first paper in 2009, I scrolled to the bottom, and it actually states, Cataline Carrico and Drew Wiseman are funded by the NIAID department of the NIH to do mRNA gene editing therapy. That's exactly what these COVID-19 shots are. This is back in 2009. Anthony Fauci, is he a big part of this, big player in this thing? Absolutely. So all I wanted to know then was, where does snake venom show up in this research paper? Because that was the search criteria. So I went to the methodology, and what they were doing was mRNA gene editing therapy research starting in 2009. And in the methodology, they explain that to cleave or cut RNA and DNA, they use a substance called snake venom phosphodiesterase to cleave RNA and DNA to hopefully insert an mRNA of their choosing into that DNA or that genetic material. Every research study from 2009 until 2020, when these vaccines went out, all of their mRNA gene editing therapy research papers funded by the NIH and Anthony Fauci all disclose they use snake venom phosphodiesterase, which is a component of snake venom that tears down the nuclear material in your body of the prey. It pre-digests the prey's tissues so that when the snake eventually gets to you and swallows you, the digestive process has already started. That's the substance they're using. And that substance has every known published side effect that you're seeing from the COVID-19 vaccines. So my concern is the people credited with creating these shots have used snake venom in their research to do mRNA gene editing therapy, and it's in their research papers. They're being funded by the NIAID department at the NIH to do it, and those shots are creating elevated D-dimers in 60% of reported medical doctors' patients. You better start looking for the snake venom poisoning the medical literature states can be a cause for elevated D-dimers to cause those blood clotting.
What they've been really plowing money in is how do you use venom to kill cancer? How do you use venom to cure heart disease? How do you use venom to adjust pulses? You know, both for every poison there's an antidote. For every venom that somebody's heard of, they should know that the other side of it exists, which is called a lectin. We produce a range of lectins. We wouldn't be functional without them. And so suddenly it also took me into this science now that I think will become the next step of my discovery process, is that there's a direct link between severe outcome of vaccine injury and of what I call spike protein syndrome, and what your body is doing with lectins. And too much lectins in your body is problematic, and too little is problematic. And lectins are a major new part of understanding how our immune system works. The science around lectins and how it triggers different immune responses. For example, it's the lectins driving the inflammation. It's the lectins driving what people are now known as a cytokine storm, that are really a huge part of what we would call spike protein syndrome. If you have something that's happening to people where the out of ordinary is thrombotic events, and in layman's term, clots. And then you find that scientists have discovered that the thing that you said causes disease causes clots. Then why is that not just a dire moment? Why is that not a moment where you say, well, of course there's going to be thrombotic events?
Myocarditis from the vaccines is a published side effect of envenomation and anti-venom therapy, which is the antibodies that are created against the venoms. That is a published side effect. Since the 1800s, by word, myocarditis is a known side effect of envenomation or the treatment for envenomation. We have actually had doctors from South Africa reach out to me after exposing my concerns about weaponized venom, calling it COVID, and venom in the shots. And these medical doctors have actually gone on interviews with me and supplied their own blood samples and their patients' blood samples and looked at it under a microscope. And what they have been looking at for a year and a half, they could not answer when they went online to see, is this what snake venom looks like under a microscope in the blood of a human? It's identical to what they were seeing in their COVID-19 vaccinated patients. These little green crystal-like structures are venom peptides, and they found them in all of their injured mRNA COVID-19 patients. And they were collecting their blood samples and looking at them under a microscope, and they were noting all of these green crystal structures, but they had no idea what they were looking at because no one told them that there's a possible venom aspect to go look at. The moment they started plugging that in, they started to realize this is exactly what published research papers show is what snake venom looks like in the blood of a patient. Then they went further and took the Pfizer vials, put them under a microscope, took pictures, and you could see what looks identical to snake venom organoids in the Pfizer vial. You do not need a snake to produce venom. For years, they've actually been extracting from the salivary glands where the snake makes the venom. In their venomous salivary glands, they can actually take cells out of the venom gland and put it in a petri dish, and it will sit there and just generate venom while in the petri dish. You don't even need the snake attached to it. It just sits there and makes venom over and over and over and over. It's sitting there becoming more and more toxic with venom if the organoids are present. When you look into antiquity, in the history of all mankind, the most utilized and most original biological weapon of all time is snake venom. And we as humans have used it as a poison. We've even thrown snakes into ships that had pirates on it that we wanted to kill. We would actually throw the venomous snakes up there. We would use those venoms as weapons. No different than the venoms on poison dart frogs. We've used those for years too. I mean, venom we have used is the oldest biological weapon.
Up to now, bioweapons are on a curriculum of people like me. Bioweapons would get an hour's worth of time at a conference because the issue with bioweapons has always been bioweapons are not viable in war because how do you not get your own? Right? How do you use a bioweapon without taking out friendlies or your own? So when you suddenly realize that the technology sped up and that you can have something that's loaded genetically and that it can only hit a certain population based on their genetics, now there's a real concern that they're taking this venom and they're putting it into an injection and then they're injecting, whether it's a child, an adult, whoever it is. And I think the part that infuriates me, and I use the word infuriates because people are being maimed and killed. And the specific moment for me, the big moment of, wow, this for me proves that this is more than likely a bioweapon, is that it matches what's known as VX.
VX nerve agent is the most deadly chemical weapon ever produced. It was accidentally discovered in the early 1950s in Britain while two chemists, Ranajit Ghosh and J.F. Newman, were working on a new pesticide to kill insects. Beginning in 1959, the United States Army began volunteer testing of VX on humans. It was found that only about 10 micrograms is more than a lethal dose of VX on an average human adult male. To serve as a deterrent against the Soviet Union, the U.S. began producing mass quantities of VX in 1961 at the Newport Chemical Depot in Indiana. Approximately 4,400 tons of VX nerve agent was produced during its production run, enough to kill every single human being on the planet a few times over.
And VX translates to venomous X agent, a product of World War II, a product of the Nazis that is currently, we have dismantled our stockpiles because of the biological treaties, and VX was a nerve agent in the family of sarin, okay, and more lethal than sarin, and tested post-World War II. In fact, there's a really interesting case around a young soldier that in the UK, they tested this on him without his knowledge, and he died. And it's a very sad story that he took up the opportunity to go and have this test being done on him to pay for an engagement ring. And then they killed him. VX is so highly, and I'm going to use the word transmissible, but that's not the way, because we use transmissible in the way that something replicates and then someone else can get it. But it's so highly toxic that you brush against the person who's been contaminated and you have a problem. So they sealed him up in a metal box and buried him. Okay, because they knew that the dose they'd used that killed him, he was now, just like radioactive material, that he could kill somebody. He was literally contaminated. Right? And then they hand him back to his father to bury this guy, and there was an inquiry into this out of the biological labs that were doing this testing post-war, and then finally the truth came out. Yes, they had killed him by mistake. I think the family probably got, a couple of pounds. Sorry, we killed your son.
What our next concern is that the conotoxins, for example, which are the snail venoms found in COVID-19 patients also, conotoxins, right now on the U.S. Department of Justice's website, they have an article titled "Conotoxins, Biological Weapon of the Sea," is the title of the article from 2012. And they state in there that there's enough of this conotoxin in powder form in factories and biological weapons facilities around the world that our Department of Justice is concerned that it could be stolen and be used by terrorists as a bioterrorism weapon. They believe this snail venom that's in a powder form in barrels or whatever they store it in, they said they know it can be stolen, taken, and then be dispersed in the air from airplanes, misters, in heavily dense populated areas. And conotoxins, when you inhale them, create respiratory arrest and cardiac arrest. Our concerns are, and Dr. Tal Brown will speak to more of this, he is in the job, his career is to identify and prevent mass killings using biological weapons, and he's aware that the real threat is to those who are vaccinated right now, that they are going to aerosolize venoms right now. In fact, he told me, I didn't even know this. Did you know you can go online right now and buy king cobra venom in an aerosolized can? Like, you can just shake it up and spray it. Now, I'd like to ask anybody on the planet, what would be the benefit of manufacturing king cobra venom in a can to spray anywhere? This has been an orchestrated, planned biologic weapon release on the entire world with one sole agenda: make people sick, let some of them die, scare enough people to get a vaccine. That's the whole agenda. Why are we spending millions of dollars to have contracted companies go in to your water sites around the country? This has been an orchestrated, planned biologic weapon release on the entire world with one sole agenda: make people sick, let some of them die, scare enough people to get a vaccine.
So this is a conspiracy. We know that evidence has been buried. If a person who's in charge, who's the government's expert on infectious diseases, can lie and basically say this is not gain of function, when it's gain of function, then you're in a conspiracy.
I did not know that since the 1970s, we have been mass producing in factories, biological weapons labs, and in pharmaceutical drug factories. We have been making synthetic venoms.
If they're saying that this is an acute respiratory issue, then why am I seeing these videos coming out of China and out of Wuhan where I'm seeing a nerve gas attack? You've never been around somebody that suddenly goes into a seizure on the floor. You've never seen somebody suddenly drop dead in a street without any other symptoms leading up to that. And we never had that in the States.
My concerns is that they're poisoning the water with venoms. It's appearing to be a respiratory disease because the venoms, it doesn't matter if they get in through the gastrointestinal tract or you breathe it in through your lungs and get it into your blood. The moment it gets to the blood, it will cross the blood-brain barrier, all these venom peptides do, and they attack neurological tissue in the brainstem that controls respiration. That's how venomous creatures kill their prey.